Cell Division by Lena Parker, Caroline Coleman, and Jack Nanny.
Mitosis and meiosis are two processes of cell division. Mitosis happens in all non-reproductive cells. It happens when cells want to grow or repair. Identical daughter cells are produced. The produced cells are diploid, and there's no crossing over in mitosis. Meiosis, on the other hand, is done by reproductive cells called gametes. Four cells are produced at the end of meiosis from one cell. They are all unique from the cell that produced them. The products are haploid, meaning they have half the DNA, and crossing over occurs in meiosis. Meiosis also has two parts, unlike mitosis. This is the cell life cycle. Prophase happens for most of the cell life cycle. Growth 1 happens first, then synthesis, where DNA synthesizes, then growth 2's process, and then the mitotic phase happens, where mitosis and cytokinesis occur.
During the first mitotic stage, the nucleus fades and the chromatin condenses into chromosomes. Each replicated chromosome comprises two chromatids, both with the same genetic information. Microtubules of the cytoskeleton are responsible for shaping the cell, mobility, and attachments to other cells during interphase and disassemble. And the building blocks of the microtubules are used to grow the meiotic spindle from the region of the centrosomes.
Centrosomes.
In metaphase, tension applied by the spindle fibers align all the chromosomes in one plane at the center of the cell,
Called the metaphase plate. In anaphase, spindle fibers shorten, the kinetochores separate, which is the center of the cells, and the chromatids, or the daughter chromosomes, are pulled apart and begin moving to the cell poles. Here's one cell pole, and here's the other.
In telophase, the daughter chromosomes arrive at the poles and the spindle fibers that have pulled them apart disappear.
Cytokinesis also occurs. In cytokinesis, the spindle fibers are not attached to the chromosomes, and they begin breaking down until only the protein of the overlap is left. It is in this region that a contractile ring cleaves the cell into two daughter cells. Here's the cleavage furrow, where the two cells are physically splitting, as well as the rest of the organelles in the cell. Microtubules then organize it into a new cytoskeleton for return to interphase. Now on to meiosis. This is interphase, and it's the same as in mitosis. The chromosomes duplicate. This is prophase 1 of meiosis. Homologous chromosome pairs exchange segments in a process called crossing over. In crossing over, tetrads of maternal DNA and paternal DNA exchange segments of their DNA. This increases genetic variation.
In metaphase 1, the spindle fibers attach to each dyad pair at the
Kinetochore, and tension from spindle fibers aligns the tetrads at the cell equator.
During anaphase 1, chiasmata break apart and the sister chromatids begin migrating towards opposite poles.
In telophase 1, the cleavage furrow forms, beginning the process of cytokinesis, resulting in daughter cells that are haploid.
And unique from one another. In prophase 2, spindle formations begin and centrosomes begin moving towards the poles.
In metaphase 2, tension from spindle fibers align from chromosomes at the metaphase plate.
In anaphase 2, chromatids separate and begin moving towards the poles.
And in the final stage, telophase and cytokinesis 2, 4 daughter cells are produced, all unique from one another. In meiosis, it's almost as if two phases of mitosis occur, resulting in 4 unique cells.